In this video, we're going to talk about T-dependent humoral immunity. With T-dependent humoral immunity, we have a B cell that will recognize an exogenous antigen. It does that with its complementary B cell receptor. Here we have the B cell receptors, and these would have the variable region at the tips, and they will recognize one specific antigen shape. So that exogenous antigen or extracellular antigen will attach to that variable region on the B cell receptor. The B cell will then process the antigen by phagocytizing that exogenous antigen. So it brings it inside the cell. The B cell then packages the exogenous antigen with an MHC class II protein. The B cell will then display that antigen on the cell surface. This process is referred to as exogenous antigen processing. Earlier, near the same time the B cell found its complementary antigen from the first slide, a T helper cell with CD4 protein, they were presented with the same exogenous antigen, either through most likely a dendritic cell or a macrophage. The T helper cell then went through clonal expansion. So here we have the B cell, the T helper cells, and these are clones. So you have to imagine these T helper cells are like a search party looking for that B cell. So here are a couple different B cells with antigen bound to MHC2 class II protein. The T helper cells survey all the B cells until they find the one with the complementary antigen. So here we have the T helper cell attached to the B cell through that antigen. CD4 recognizes MHC class II. So that B cell presents the antigen to the activated T helper cell, and the T helper cell will send a cytokine signal which induces that B cell to undergo clonal expansion. Here we have some B cell clones being made. Some of those new B cell clones will differentiate into plasma cells which secrete antibodies. Others will differentiate into memory B cells. During a primary immune response, this process takes about 10 to 14 days for the B cell to undergo activation, clonal expansion, and then differentiate into those plasma cells. And then we finally get that antibody secretion. So 10 to 14 days for the highest level of antibody produced. And if secondary response, those memory B cells are responsible for a fast response, typically about 3 days, and you have a high titer of antibody in your system. The results of antibody-antigen binding, again, include agglutination, activation of complement, opsonization, antibody-dependent cell-mediated cytotoxicity, and neutralization. And again, if we have neutralization, if you have agglutination, that means that those are also being opsonized. So both of these two will go back and also cause that opsonization. This concludes the T-dependent humoral immune response video.